We have a request to all the participants before starting the session. Please do not switch on your video or audio between the session and add your comments, suggestions and questions, if any, to the chat box. The feedback for the session will be given at the end of the session and uh, through the short chat box. Hope session will be beneficial to you and fruitful. I invite Head of the Department of Biochemistry, Dr. Julie Checkup, to introduce our resource person, Dr. Anish Kumar AG, and to welcome you all. Thank you all. Over to you, ma'am.
Thank you, Dr. Asha. A very warm welcome to you. I want to extend a warm welcome to the NICA-sponsored national webinar on recent advances in DNA sequencing technology. At the outset, I would like to thank our Secretary, Dr. Vinny Verghese, and our Principal, Dr. Shandy A. Avida, whose never-ending support made this webinar a reality. On behalf of Bharat Vinicius College, Kodamangalam, I would like to extend a warm welcome to our esteemed resource person of this webinar, Dr. Anish Kumar AG, Scientist for National Institute of Immunology, New Delhi. After obtaining his PhD from Center for Cellular and Molecular Biology, Hyderabad, he did his postdoctoral research at National Institute of Health, USA. Returning home, he is now currently working as Principal Investigator, Molecular Genetics Laboratory, National Institute of Immunology. Dr. Anish Kumar has published 22 research papers in peer-reviewed journals and is a recipient of several awards and honors. I welcome you, sir, to this webinar. Next, I would like to welcome the technical support team here at Bharat Vinicius College, especially Asha Miss, Paul sir, Francis sir, Nidhi sir, Evin and Jacob for their never-ending support for organizing this webinar. I would also like to extend a warm welcome to all my dear colleagues, teachers from various colleges, research scholars, students, and our alumni to this event. I am sure you will have a fruitful session ahead. Now I would like to invite Dr. Anish Kumar for his presentation. Over to you, sir. Thank you.
Hey, thank you, Dr. Julie and Asha, for asking me to give this seminar. Um, okay, let me present. Okay, the slide is visible to all, right?
Yes, sir.
Okay, so so I thought I will talk about next generation sequencing technology. One reason is we use it in our lab a lot. So I thought it may be a good idea to introduce, it in some detail to the students. So that's why I thought I'll take this topic. But I'm not really planning to give a complete, a very detailed and complete overview of the technology, because it's actually quite vast field and extremely rapidly, evolving field. So I'm not going to do that. But what I would try to do is to give a glimpse of it, the general, the generalities of different methods, uh, and then try to give some idea about how it's used to understand the functional genomics aspects. Okay, not just DNA sequencing, but how cells behave. How we can use this technology to understand how cells behave under different conditions, using some of the work that we have done. So I won't be going into very detail of that as well, uh, but my intention is to actually introduce uh, the technology and its use in functional genomics. Uh, so this slide should be, like familiar to all of you. It's how the DNA is packaged in our cell. Of course, DNA was, you know, confirmed to be the genetic material in early 50s, and the works of Nirenberg and who found that actually DNA is, the template for protein synthesis. So, like the RNA made from DNA is a template for protein synthesis. So understanding the sequence of DNA became very important, not only just out of curiosity, but also to understand the diseases, because a lot of genetic diseases were known by then. And to understand such diseases also, we need to know what are the sequences on the DNA. Uh, and though, immediately after the discovery of the structure of the DNA in 53, like, in a few years after that, uh, Robert Hawley has found, first sequence of first nucleic acid molecule, like a tRNA. But that was a very complicated method that normally, it took 3 years just to sequence a 17 nucleotide RNA. So that is very, very complicated method, and that was not really usable in any meaningful form in most of the laboratories around the world. So apart from being, the DNA being the genetic material, you also aware about this kind of flow of information within the living cells from DNA, which can replicate, and DNA to RNA, which is transcription, then translation, then proteins, then proteins actually, metabolites, and proteins and metabolites together finally determine the phenotype, whether it is a disease, whether it is things like cancer or diabetes or a very healthy person. Everything is determined by a combination of these things. And we know that our we have like 25 plus thousand genes at least, and there are many variations, splice variation, post transcriptional variations among the genes, gene expression. So all these variations together actually determine the phenotype of an individual. And to understand that phenotype, or the interplay between the genetic variation and the variation in the gene expression, whether it is at the RNA level or protein level, and finally how the metabolites are changing within the organism, all these things combined actually determines the disease condition or a healthy condition. So looking at individual genes, okay, that's not really sufficient in many cases, especially if you take diseases like complex diseases like diabetes. Okay, it's not really genetic, but people have found there are differences. There are certain things that are present in many diabetic patients, certain DNA modifications, mutations, certain changes at the level of gene expression, certain changes at the level of protein or metabolites. So to get a better understanding of such things, we need to actually understand at a global level how these molecules are behaving. So we need global technologies. Okay, so that's an advantage. So especially the whatever I have, the red font here, the DNA RNA, that means the transcription and translation. These two processes can be followed at the bulk or at the global level by knowing the DNA sequence, or the DNA and RNA sequences. Okay, so that's another aspect that I'll come back to this in detail later. So from the discovery of DNA as a genetic material, and after the structure was published, there was a lot of attempts to sequence DNA or RNA for that matter. And interestingly, in the same year, in 1977, two groups. One was Maxam-Gilbert, and the other was from UK, Sanger. So both of them published methods to sequence nucleic acids, especially DNA molecules. So Maxam and Gilbert from Harvard, they actually used a chemical sequencing method, and Sanger used a polymerase dependent, a method now called chain termination method. And both of them shared Nobel Prize for doing, for identify or being succeeding in developing a method to sequence nucleic acid. So I will just touch upon what actually are, because it's interesting to know, the history as well. Though most of the people don't really use Maxam-Gilbert method now, I think it's better, to know a little bit about that as well. So it's a chemical method of DNA sequencing. So what they do is, you need to first make a single-stranded DNA molecule from a double-stranded DNA molecule to actually do this experiment. So you first a single-stranded DNA you make, and you have to label at the 5' end with a radio label, a radioactive P32. So there are ways you can actually add a radio labeled phosphate group to the 5' end of an RNA or DNA. Now, what this method use is, it actually, if you have one DNA that you want to sequence, you have to divide it into four different tubes, like given here, four different tubes, okay? And to each tube, you add certain chemical reagents that specifically modify, chemically modify specific nucleotides, like, a chemical that specifically modify guanines, and a chemical that modifies all purines, and same way all pyrimidines, and the one that actually specifically modifies cytosine. So so once you do that, the same DNA molecule is in the four tubes, and you add these reagents, and what this group found was, if you add another chemical to this modified nucleotides, it cleaves the DNA at the modified site. So that's the idea. Now the issue is that you have to be really careful, so that when you're adding these reagents, it shouldn't modify all the nucleotides. Like the reagent that modifies guanine, it should not modify all the guanines within the same DNA molecule. So you have to go for a single hit kinetic. That means one molecule of DNA is modified only once. Okay, so that's a key in this process, in all the sequencing process actually. Okay, so that way, then when you actually cleave them, you get all possible lengths of DNA with a phosphate group attached to the radioactive phosphate group attached to the 5' end. So once you do that, you can run a polyacrylamide gel, a urea gel, a denaturing polyacrylamide gel of very high percentage, so that now you can separate these fragments that are created. Okay, so you run them in 4 different wells, one well for each reaction, and this is how you generally read that gel. Okay, so this is the full length one, radio labeled full length one will be the top band here, and for this particular sequence, ATG, ATC, ATGC, ATGCA, here you will have A here. So that means, so you will have the band only in this particular well, A plus G reaction. Okay. And when if the second nucleotide is T, you will have a band only here, T plus C. Okay. On the other hand, if you have a G, you will have a band at G and A plus G. So both, this G is a purine, so both places you can find a band. So basically what you have to do is you have to get an autoradiogram, you run the gel, you expose it to X-ray sheet, and then you have to sit and read it like this. Like if you have a band only in the second well, that means it's an A. If you have a band in first and second wells, it's a G. If you have a band only in third well, it's a T. So like that you have to sit and read. So you can see it's actually quite difficult. It takes a lot of time, and obviously it is not really great for if you want to do it at a large scale. So that is one biggest disadvantage of this. And another thing is almost all the reagents used in this method are extremely toxic. Obviously, you can understand if it's modifying DNA, means most of them are supposed to be carcinogenic, right? So that's the point here. So and also to make a kit, if you want to really use it at a very in a commercial basis or in a wide usage, then you need to make kits for it, right? And that's going to be extremely difficult because these are finicky reactions with extremely toxic reagents. So that's where the second method, which was introduced by Sanger, and you will see that this method has prevailed all throughout. The whole human genome was sequenced using this method, uh, and even the many of these NGS platforms, the next generation sequencing platform that we'll talk about later, they also use some extent of some modifications of this method. So this became really the popular DNA sequencing method. So most of us, when we say DNA sequencing, we actually mean Sanger method. So it's a method where we use special nucleotides called dideoxy nucleotide, ddNTP. So you know what is a dNTP, which is a normal nucleotide in the DNA, which is the bottom one here. So which doesn't have an OH at the 2 prime. So it's a 2 prime deoxy NTP. Now dideoxy doesn't have uh, an oxygen at the 2 prime and 2 prime and the 3 prime positions. So the problem is, if you're making a DNA, so you must have already studied DNA replication. So it's always 5 prime to 3 prime, right? So this 3 prime end, this 3 prime OH, the 5 prime phosphate of the next nucleotide comes and attacks. So here in the case of dideoxy, there is no oxygen, so that's not going to happen. So whenever an elongating DNA chain, a DNA polymerase incorporates a dideoxy NTP, it stops there because it cannot add another nucleotide there. So it's called a chain termination. Okay. So again, it kind of looks very similar to the chemical method. Like that, we need to have 4 different tubes here, but here we have a labeled primer. Okay, the label can be radio label, where you can actually run a gel like this and do the autoradiogram just like we have seen in case of the chemical sequencing method, or it could be fluorescence. So many of the semi-automated sequencing machines used fluorescent labels. So that's an advantage of this method actually. So you can use a primer that has a label, can be radio label or a fluorescent label, and you set up 4 different PCR reactions. It's simple PCR reaction, and in the only difference between these 4 tubes are one has dideoxy G at a lower concentration. It has deoxy G there, plus you add a little bit of dideoxy G, and same with DDA, DDT, and DDC. So how it works is, there is the deoxy G nucleotides there, so normal PCR happens. But when it happens, a small fraction of the products will have they'll be incorporating DDG instead of DG. Same way DDA instead of DA. So in those fragments, actually they stop the reaction there. So again, what we get is a wide range of lengths of the the molecule, DNA molecule. And again, as I said, we have to run the gel. That's one method to do that, either with a fluorescent label or with a radio label. And again, we read like this. But unlike the previous one, this is more definitive. Like, you have A means you have a band only in DDA added one. If it's a T means you have band only in the DDT added one. So like that. So it's much easier to read. So that's one advantage of this method. And then soon enough this was automated, because you can run a fluorescent label primer, okay, so that the gel can be scanned by a scanner, and easily the positions could be read by the computer. So that was one method. And there is another modified version of this is, you use fluorescent labeled ddNTPs, okay, and run the reaction. Again, you have to have 4 different reactions, and finally what you do is you run a capillary electrophoresis. Okay, capillary, it's a what is a capillary electrophoresis, right? Like plate electrophoresis, you have the gel in a capillary, and when you do a capillary electrophoresis, the the color, the oligonucleotides get separated by their size, and each one has a particular fluorophore attached to it. So there is a scanner here again. There is a and a photo receiver here, so it can detect whenever each color pass through. Ideally, you will get this kind of a chromatogram here. So based on that, you can again find the sequence. So this made a big deal because actually you could, the capillary electrophoresis is much faster than the normal plate electrophoresis. So you can actually run it faster and you get much accurate readings as well. So if you send out, if you do a cloning and you want to confirm it by sequencing, you send it out for sequencing. They do this method, the capillary method of Sanger sequencing. And most of the DNA sequencing for Human Genome Project was also done using this method. So it's a very powerful. We actually learned. So yeah, this again, a depiction of the Sanger method. So if you're doing, a de novo sequencing of a genome using Sanger method, so the general procedure is you have to fragment the DNA and then you have to clone it into a vector and then put it into bacteria to amplify the vector, then isolate plasmids, and you have to actually sequence each clone, or not each clone, but a variety of clones. So it takes a long time and a lot of effort and a lot of money to do this whole procedure. Okay, so that's a major drawback of this. If you want to get a genome sequence, it could take years actually if you want to sequence a genome de novo. So that's a big problem. So that's when in last few, like probably 10 to 12, maximum 15 years, I guess, new methods have come up which actually use massively parallel sequencing of millions of DNA molecules. Okay, so in case of Sanger sequencing, you have to make these clones and you sequence one clone at a time. Okay, so that's what you have to do. So on the other hand, in this new method, it was possible in one go you can actually sequence millions of molecules. Okay, so it was actually a technological advancement probably of this millennia. So the basic nature, how we can actually do this, sequence millions of molecules at one single go. Okay, so the basic idea, or the core of all these methods, though their chemistry is different, their methodologies could be different, but one of the core thing is shown here. Like you have a chip, okay, a glass chip, a special quartz chip, or any chip, and in this chip actually you can make clusters of identical DNA molecules. Like you can, like here, very few clusters. Each white spot is a cluster of DNA with identical sequence. Okay, so you have one molecule of DNA and you will have one cluster made of this. Now this cluster will have thousands of DNA molecules of the same sequence. Okay, and a computer or a CCD camera can actually distinguish these spots. So I will show you an image of this later, so then you will understand this better. So then once you have these DNA clusters, now you can add either individual NTPs or fluorescent labeled NTPs, and see if you add a fluorescent ATP labeled with red fluorescence, how many of these spots turn red. When you add guanine with the green fluorescent, how many of these spots turn green. So like that you can actually build a sequence. It's nothing but a chip and a scanner, a laser scanner. That's the crux of this method in all these methods. So and the methodologies, how to make clusters, that vary with the technique. Okay, but in general there are, okay, I'm sorry. Yeah, there are several platforms are available. I have just listed 3 here, but actually it's not just 3. There are many other platforms available for doing next generation DNA sequencing. The first one ever to come up was called 454 pyro sequencing. Okay, So I will, since that's the first one, due to the historical importance, I will introduce that. The most widely used ones is Illumina. So that's what the most, most of the people use this particular platform, Illumina. So I will introduce what actually happens in this as well. Solid is also a good model, but I'm not going to talk about that because of we may not have enough time to talk about all these. So, but in all these uh, methodologies, there are certain things that are common. I already told you about the scanning method of the chips. Okay. So, but other than that, the sample preparation. So we have already seen, if you want to do a de novo sequencing, you have to actually clone all the fragments. You fragment the DNA and then you have to clone all the fragments to get an idea. In this case, there is no cloning is involved. So you have the DNA, the total DNA, you fragment them into smaller pieces, much smaller pieces, like, much smaller than what you do it for a big, quantic sequencing with Sanger sequencing. And so you have all DNA in a tube, then you, fragment them and you attach a pair of adapters to either end of these fragments. Now what you have is, the whole DNA is into small pieces and each piece has the same identical sequence there. Okay, the adapters have identical sequence. Every single piece has the same adapter sequence attached to it. Okay, so that part is kind of common for most of the next generation DNA sequencing, or I would say second generation DNA sequencing. This is common, and once you have this, then you make the clusters. I already told you there are clusters, right? You have clusters of DNA on chip. We will come to that in the next slide. And once you make clusters, then you have then you do several cycles of sequencing. That means, if it's a, depending on the chemistry that is being used, you use a polymerase to add a nucleotide, then wash away, unbound, then you scan. That is a cycle one. Okay, then you get this kind of a, the cycle one, a picture. You have different spots here. Okay, each one has different color, so the computer will record that color. Then there are ways to get rid of the fluorescent label, and then you add the next nucleotide set and you see what's happening here. So it's changing. In the first case, the spots are G, A, T. These three spots are G, A, and T. The second one, it is T, G, and G. So that means these are the two. You get a dinucleotide sequence there. So like that, you can go for any number of cycles, and finally you get the sequence. So that's basically the idea. Now, I told you about generation of clusters, right? So that's an interesting, concept and a major technological advance again. So in sequencing systems like 454 pyro sequencing, solid, and most of these sequencing platforms use a particular method called emulsion PCR. Okay, so it's a very interesting methodology. So I guess, I mean, I'm sure most of you know all of you know what is PCR, right? You take a piece of DNA, you give two primers, and then you give a polymerase NTPs and do the cycling. So you'll get tons of amplified DNA product from that. So emulsion PCR is essentially the same, but it does in a slightly different manner. Okay, it uses some certain kind of beads shown here. These beads that are attached to certain oligos. Okay, these oligo sequences are complementary to the one of these adapter sequences here. Okay, so we have already fragmented the DNA and attached the adapters to the DNA, both the ends of the DNA, right? One end adapter is now complementary to the oligo that is attached to these beads. Then you actually enclose this whole thing in an emulsion, in a droplet kind of thing, basically. Okay, so this droplet practically has one bead that has these adapters, these oligos attached to it, and one DNA molecule. And within this droplet, we have all the other reagents to do the PCR. and the PCR will be done. So each cycle, this DNA molecule, one DNA molecule, it will get denatured, it will, and then anneal to one of these fragments, and the PCR the amplification will happen, and then denature again. It will go to the next one. Like that, ideally, you will get a bead full of identical DNA sequences. So to start with, you had one DNA molecule. Now you have a bead that has probably thousands of DNA molecules of the same sequence. You just amplify the same oligonucleotide there. So that's one cluster that you have. Okay. Now, in case of pyrosequencing, it's a very interesting chemistry that's happening here. And again, it's a first method that first methodology for NGS, the massively parallel sequencing that was discovered. So once these beads are there, okay, with all the attached DNA, amplified DNA, that cluster, so you put them into a chip. Okay, it's again a chip, but here the chip is microwells. It's a very minute, picoliter kind of, volume, very minute wells, where exactly one bead can fit in. Two beads cannot fit in there. So it's that kind of small, wells. So then you add these beads to these wells, okay, and then you add all the reagents required for that. So now what happens is, when then you add one nucleotide at a time, like you give DGTP or DATP at a time. So first you add, flood this whole thing with the GTP. Let's talk about DGTP. And for whichever wells, there is a primer here, right? So whichever well the next nucleotide is to be added is a G, it will add a G. So if you like you have studied replication, an DNTP is added, a pyrophosphate is released, right? So same thing is happening here. A pyrophosphate is released within this well. Okay. This reaction also has, another reagents, like which is called, adenosine phosphosulfate, and an enzyme which actually you use the pyrophosphate and APS, the adenosine phosphosulfate, to make ATP. Okay, so this PPA released will lead to formation of ATP within these wells. Okay, and these wells also have luciferase enzyme there. Okay, and its substrate there. So luciferase is, firefly luciferase, but you have the enzyme that is there in the fireflies, that makes light in fireflies, right? The same thing. So this luciferase plus ATP, that gives light. So you add GTP, you flood the system with the GTP, and if this well has, the nucleotide to be added is G, it will add G, it will make pyrophosphate. That pyrophosphate plus APS will give you ATP. That ATP plus luciferase will emit a light from this well. Or if, and the adjacent well, if it doesn't have it, it's not adding G, there is no pyrophosphate and no light. So there is a scanner on top which actually looks for this light emitted from each well. So if it's emitting a light when you add GTP, it says that, okay, yeah, there is a G there. Okay, same way C will be or D will be. In case of ATP, it is a slightly different, because you cannot add ATP there because of, ATP can be, or even you don't want any issue with the ATP that is required by luciferase. So you are adding a particular variant of ATP here. So individually you add these nucleotides and then scan the chip. Finally, and after each step, you wash it with epirase. Epirase is an enzyme that actually kills all the remaining ATP, unbound ATP or NTPs. Okay, so there will not be any background. So this is what actually, it sounds complex, but the beauty of it is it's easily automatable. You can completely automate this, and it can be happening at the million molecule in a chip scale. So that was the beauty of this method. Okay. So another method, as I said, the most widely used method is the Illumina technology, which uses very similar method like Sanger sequencing, for the sequencing reaction, but here the cluster generation for it is slightly different from other sequencing methods. Like, it's not using the emulsion PCR, it's actually using a bridge PCR. So what it has is, a chip that has a large number of these oligonucleotides planted on the chip surface. Now you have your DNA, okay, with these adapters, right? And whatever oligo what is there on the chip is complementary to these adapter sequences. So when you denature and add your DNA there, those your DNA is going to anneal to these oligos that are stuck on the or planted on the chip surface, okay, and then you do PCR basically. So when you do PCR, uh, sorry, when you do PCR, it actually makes a double-stranded DNA, okay, and then it can it can actually, you can see it can bend over and bind to the next nucleotide actually, the other adapter there, and that can again initiate another PCR cycle. So like that you keep on, and after that you denature it, it goes up again, and finally you end up having a cluster like this. You fill up the whole thing with the clusters. So this is the method that is used by Illumina for making clusters. So basically each cluster now has again thousands of DNA molecules of same length. So you start with one molecule, your library has one molecule, and now you have thousands of molecules in one spot. Okay, that is required because that's how you actually increase the intensity of the signal. Because when you're adding a fluorescent label dye, just one nucleotide, it will not be that visible. But now all these 1,000 molecules in a cluster is going to add the same dye, right? So that will come up as a bright spot there. So that's the idea behind this method. So ultimately, the idea, and also they use specific nucleotides. Okay, they use a mixture of, like in previous ones you add one nucleotide at a time. You add GTP, you add DATP one at a time, and then wash out. Here you can add all of them together because each one has a different fluorescent dye added to, attached to that. Plus their 3' end are blocked. Okay, the 3' OH is blocked. So it can add only one nucleotide at a time. Okay, uh, so you add this, the polymerase will add one nucleotide, then it cannot add because of the 3' blocking. Okay, and each one is fluorescently labeled. Once that step is over, you scan the whole slide with the 4 different filters, okay, based on the colors on each ATP. So you get this kind of a picture, spots. So each spot here is a cluster. Okay, so each spot here is a cluster. Like if it's yellow here, that means it's this cluster. It has T here. Here it is blue, means probably G. Here it is A, like that. I mean, I'm just arbitrarily saying that, but I hope you got the idea. So this is how the combined one look like. So the computer actually stores that, okay, yeah, this particular cycle, this port has yellow. Now next step what you do is, both the fluorescent label and the blocker are reversible. They can be cleaved off. Once you cleave them off, that DNA will not be fluorescent any longer, and the blocker is gone. So it can add another nucleotide there. 3' OH is free now. So you remove the blocker and the fluorescence, and then you add the next, go for the next round again with the same batch of same combination of NTPs. Okay, so then after that, then this may turn out to be purple. So that would say, then the computer, okay, this spot, previous one it was yellow, now it is purple. So the next nucleotide is this. So that's how the machine makes the calls. Okay, so this is basically the technology part of it. So this is actually most advanced one. You can actually get billions, not even millions. You can actually get billions of reads in one go. You can finish a genome probably in one run. That kind of capabilities are there, and it takes probably 1 or 2 days to run the whole thing, but it's quite fast, and it's much cheaper also nowadays. Okay, and there are other platforms as well, like there is one called Ion Torrent, which actually, again, similar to what we have seen in pyrosequencing, it actually happens in wells, but it actually measures the pH. There is no scanning for the fluorescent level. So each nucleotide addition leads to release of one hydrogen atom, which decreases the pH. So basically the machine does is it look for pH. So there are other methodologies. So we are not going into the details of that. So so why do we use this? We all, every time we hear about NGS or sequencing, yeah, we can sequence a genome, $1,000 you can sequence a genome, everyone can get our genome sequenced. Like India has this 10,000 genome project where 10,000 of people are going to be sequenced, whole genome is going to be sequenced. So why do we do this? So the idea is, now we know what is we have a reference human genome, but all of us know that's not same for all, right? There are tons of variations between people, and some of these variations are associated with the diseases. And by sequencing a large number of people, we can identify if we can actually connect or find correlations between variations and the diseases. That will give us a better understanding of how to treat the disease, like personalized medicine. Okay, and another important aspect is functional genomics. uh,
Excuse me, sir.
so that's actually our, yeah.
Sir, it's not visible.
Yes.
This PPT is
Oh,
not
it's not
visible.
visible. Oh, it, okay. It was visible before, or it just?
Yes, it was. Just now, just
It was. Is it working now? No.
Not yet, sir.
Not yet. Okay, let me stop the presentation and re-share it then.
Okay, sir.
Okay. Okay. Is it working now?
Yes, sir.
Okay. So there are so much of research happening in terms of medicines and finding new disease associated mutations, variations, polymorphisms, everything, by simply looking at the DNA sequence of tons of people. Okay. Another aspect is functional genomics, which is basically our interest. So how an organism responds to a particular condition, okay, disease condition or a stress condition. Okay, it's like before and after stress, there may not be a difference in the genome sequence, but we do see that there are a lot of differences happening within the cell. But we don't really understand what are those changes. So one of the easy ways to understand is to look at how the transcriptome changes. That means how the mRNA levels are changing. There are differences in particular mRNA levels, and we cannot do this one by one. We can do the complete whole global sequencing, and we can get a global understanding of the transcriptome by doing a complete transcriptome sequencing. I'll come to that. So basically, functional genomics is, it's a combination of, it's a study of how genome, epigenome, transcriptome, proteome, and metabolome work together to produce a particular phenotype. So we all have different phenotypes, and our phenotypes sometimes change when we get a disease. Like now we will be healthy. Uh, Now we'll be healthy, but after we get disease like diabetes or high BP, that's a different kind of phenotype. Okay, these are associated with the differences, the global differences, the network of the genes and their mRNAs and the proteins and the metabolome or metabolites. They together actually brings down this kind of a phenotype. Okay, so genomics and epigenomics. Genomics is the mutations in our genome, which we actually either got from our parents or we acquired somatically. It could be, and epigenome is the modifications on the DNA or on the chromatin. Chromatin is made of histones. Okay, so that's epigenomics. So that can affect gene expression. Transcriptome is the study of RNA molecules present in a sample. Proteomics is basically the proteins. What are the proteins? And finally, metabolomics. Okay, out of this, genomics, of course, the sequencing is important for that. Epigenomics also, the DNA sequence is very, very important. And transcriptomics, that means you are looking at the mRNA levels. You can actually convert the mRNA into cDNA and do the sequencing reaction on the cDNA. So that's how you do the transcriptome analysis. So that's mostly the focus of our lab. So basically genome. So this I already told. So go to the next slide. So one part of our lab work on MTB, Mycobacterium tuberculosis. Okay, it's a very, 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 India has a huge burden of MTB with tens of thousands of people dying every, year. Like, for example, in 2017, when you look at simply look at the numbers, in 2017, you have had around 10 million new cases, in 1 year of MTB, and 1.6 million people died. I don't know the math. I mean, I don't exactly remember what is today's COVID, statistics is, but this is MTB happening like almost every year. It's going on like this for decades actually. So it's a very, 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 very bad disease. I just wanted to put it into the context of COVID as well. So one aspect of MTB is, you get the disease from another person through, aerosol. Okay, and once that person, once the bacteria gets into the lung, those bacteria are taken up by macrophages, alveolar macrophages. Normally, macrophages are the cells that kill bacteria. They phagocytose the bacteria and they kill the bacteria. So that's their normal function. But in case of MTB, they live in macrophages. That's their natural host cells. So the macrophages cannot kill them, but they initially what macrophages do is they cannot kill them, so they recruit other immune cells. Finally, forms a granuloma. Okay, a granuloma means that actually it's a very fibrous tissue which encloses the whole or mycobacterium cells inside. Okay, and so that the bacteria doesn't get any oxygen, any nutrients. So it's kind of containing them. It's not killing them. It's actually trying to contain them. Whatever the bacteria it couldn't kill, it's trying to contain them. So normally, sometimes they get eradicated just like that. You end up with the calcified granulomas. That's okay. And in most cases, like in majority of the cases, like 90% of the cases, the granulomas remain there, for years. Okay, without them, like it's supposed, almost 25% of the population is supposed to be infected with MTB, and most of us, have them in this particular form, in a latent form. They're not. That's not disease causing. Now, in 10% of people who have who were infected with MTB, so they actually, the bacteria somehow start, replicating within the granuloma, and the granuloma will burst, and finally that forms the active disease. Okay, so that's how the cycle is there. Yeah. So so there are several, the latent TB is actually the major hurdle in eradicating TB, because you don't know who has the disease, right? If once somebody has a disease, active disease, that person has a disease, you can treat them. But there is no easy way to actually find who has the latent TB otherwise, because that's inactive. So people do not have the disease. Now, I already mentioned the function of macrophages is to kill, the invaders, including bacteria. Okay, so how it does is, when there is a bacteria, macrophage actually, phagocytose them. Okay, once they are phagocytosed, actually immediately there are oxidative stress and nitrosative stress. That means, ROS, the reactive oxygen species that can damage DNA, and same nitrosative stress, reactive nitrogen species, which can also harm DNA and also the other molecules in the cell. and then it is going into lysosome where the cells are getting killed by the low pH, acidic pH. So that's how normally bacteria are killed by macrophages. But macrophages can actually survive these conditions, which actually kill most of the other bacteria. Okay, so that's the question. So I won't go into the details of that, whole detail. My intention here is to just to show one of our research question, how it is relevant to the functional genomics aspect, and give an idea about that. So the question asked is, how does transcription and translation correlate under stress? So that's basically we are asking. So you know that in bacteria, like transcription and translation are coupled. At least the first round of transcription is coupled with the translation, right? That's a normal molecular biology 101 that Mr. had in the UG courses. So then the question is, once these transcripts are made, are all of them getting translated at the same level? Okay, so you have RNA. We look for the RNA levels as a proxy for gene expression, but it has to make proteins, right? Whether all the RNA are making proteins or not, or are they making at the same level? How is it being regulated? So that is basically, and when you actually shift the cells from normal growth condition to an oxidative stress condition, okay, how does this change? Whether there are the mechanisms which immediately activate some genes, translation of some genes, while the transcription is not that affected. So that's kind of questions that we are trying to address. So we had to do two experiments for this. One is RNA-seq. Okay, RNA-seq, the total RNA-seq. So it is basically the same NGS method that we have described till now. So you take total RNA instead of DNA. Here you are taking total RNA. Okay, so this picture is actually for eukaryotic mRNA, which you can see probably a tail here, but just ignore that part. But otherwise, everything else is same. You have an RNA, mRNA, not one RNA. You have every single RNA in the cell. Now you take that RNA and you fragment them. Okay, you fragment them using one of the several methods, and then you attach the adapters to that. Okay, once you attach the adapters to that, and then you do the sequencing. We use the HiSeq, the Illumina HiSeq 2000 platform. Okay, you do that, and then you get millions of reads, like 10, 20, 30, depending on the depth that you have selected, millions of reads you get. So these reads are DNA molecules, right? Like you have varying from depending on the reaction that you have used, you have sometimes 100 nucleotide long reads, sometimes 35 nucleotide long reads, sometimes 50. So you have these reads. Reads means DNA sequence. Okay, you have millions of this DNA sequence. There are computer algorithms to map these reads. So now we have to find, right, where each of these sequence is mapping. So there are algorithms that actually map these reads to the genome. So now we have a genome sequence, right? The MTB, and like human or mouse or yeast, MTB genome is also been sequenced, and the genes are already demarcated. Like in the genome, position X to position Y is this gene, gene A. Position A to B, it is gene number B. Like that, it has been annotated. So this particular program will see, okay, these reads map to this gene, these reads map to this gene, these reads map to this gene, like that. So then we can actually make a count. Okay, how many reads are mapping to this gene, how many reads are mapping to this gene. So if your RNA is abundant, okay, you have gene A, which has only 10 reads mapping to it, okay, and you have gene B where 100 reads are mapping to it. That indicates that gene B is its RNA levels are pretty high, because the number of reads normally depends on the abundance of the mRNA in the cell. So that's how you generally do the experiment. You basically count them for each gene, how many reads are there. Okay, that's used for the next analysis. So that's for RNA-seq. So this will tell you what are the RNAs. But our interest is actually to understand how many of these RNAs are getting translated. So how do you do that? Again, this is similar to RNA-seq, but it's a footprinting experiment. But what we do is, it's called ribosome profiling or RiboSeq. See, the translated mRNA, the mRNAs that are getting translated are bound by ribosomes, right? Okay, and if you go back to your textbooks, you will see that each ribosome can actually protect, okay, or it binds to around 28 to 35 nucleotides of RNA. Okay, now if a particular mRNA is translated at very high level, they have large number of ribosomes attached to that. If one is not translated well, it will have less number of ribosomes attached to it. Okay, so that's how it is. Now, what we do is we simply digest all the RNA with RNAs. So everything gets digested except the RNA protected by the ribosome. Now we isolate that RNA and do the next generation sequencing, similar to the RNA-seq that we have described, and then map them onto the gene. So now what we have is we have to compare riboseq and RNA-seq. So we have, we know that in RNA-seq, okay, how many reads map to gene A. We know that RNA-seq, how many reads map to gene B. We also have now in ribosome profiling or riboseq, how many reads map to gene A and how many reads gene maps to gene B. Okay, if transcript levels are completely driving the translation, you will have same ratio between RNA-seq ratio of RNA-seq reads in A and B and RNA-seq reads in between A and B in the riboseq. Same ratio you will get. But if, like, say, a particular RNA-Z is there in the transcriptome, that means there in the cell, but it is not getting translated. In that case, you will RNA-seq, you will see a lot of reads mapped to that gene. Riboseq, you will see less number of reads mapped to that gene. Okay, so we will actually find what are the we will found the what are the differentially expressed genes. That means under the two different conditions, oxidative stress and normal conditions, what are the mRNAs that are differentially expressed. That means which mRNAs are upregulated, increasing under stress condition, which mRNAs are decreased under stress condition. Okay, that's differential expression in both transcriptome and translitome. That means RNA-seq and riboseq. Okay, so this is how it looks like. This is the riboseq data. So normally, most of the reads, maps pretty well. They are in this diagonal line here on the x-axis. What you have is the read counts in log phase, and on the y-axis you have on the oxidative stress. Okay. So most of them, the blue dots, which are close to the diagonal line. That means they are changing in a very, 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 very correlative manner. But the pink ones are, you can see, they are on the surface. Okay, so those are the ones that are upregulated in oxidative stress. So they have higher number in oxidative stress and lower number in log phase. Same way, RNA-seq also you get similar profile. A large number of genes are upregulated in the oxidative stress, which is the CHP RNA. Okay, so this is how you actually how the data looks like. The pink ones are the differentially expressed or upregulated genes. Okay, so now what we asked was, if you look at the upregulated genes in riboseq and if you take upregulated genes in RNA-seq, how many of them match? If increased RNA levels completely determine the increase in translation or riboseq levels, it should be same. Both should be same. So that's what we looked at, but it's not. So you see around 51 genes are specifically enriched in riboseq compared to RNA-seq. That means those are, translationally upregulated, not transcriptionally upregulated. Okay, and so we get a bunch of genes now. Okay, I'm not going into the details of the individual genes, but this is how it works. You get when you do this analysis, you get a large number of genes, names of or the accession numbers of large number of genes that are upregulated or downregulated. So now what do you do with this? You have a list of genes. You have to make sense out of it now, right? So once you have that list, you again use that list against different databases. It's called pathway analysis or pathway analysis or gene ontology analysis. What you do is, you have n number of genes, like you have 100 genes. Okay, if a particular process is upregulated in the stress condition, okay, the genes associated with that process will be enriched in those genes. Okay, that's basically the idea. So just to show an example, this is what we find in case of the genes upregulated in oxidative stress in MTB. Okay, 23.5 percentage of genes. Okay, we know that they are part of cholesterol catabolism. Okay, like that 11 percentage are associated with other lipid fatty acid metabolism. So that means, and also almost 10 percentage of the genes are associated with oxidoreductases, dehydrogenases. So those are important for alleviating the oxidative stress cause damages. So those are upregulated. That makes sense. Plus cholesterol catabolism. That is, so basically what you get is, now when a cell is shifted from normal growth to oxidative stress, what processes are more important for the cell. So in oxidative stress, it seems cholesterol catabolism is very important. So this is how you make sense out of this data. Obviously, we will, we have to then go ahead and prove these by doing other experiments. But the idea is, this is how you get an idea about how, at the global level, how you understand the changes in processor. You are not really looking at individual genes any longer. You are looking at processes. Like now we can say that when the cells undergo oxidative stress, you need more oxidoreductases, which makes sense because you need to alleviate the oxidative stress damage. You need iron sulfur metabolism because iron sulfur complexes are required for oxidative stress response. So that's how actually we get an idea from this kind of functional genomic studies, what are the important processes under stress conditions. Okay, so I think I will just stop with that. It's almost time. So these are my lab members. These are the PhD students: Gagan and Amika, Neha and Chen. Then Somdev is a JRF, Smita is on SRF, and Kim Singh Ji is the technical officer here. And these are the collaborators. And lab is funded by DBT, DST, SERB, and NII. Yeah. Questions?
Hello.
Hi.
Hello. The session
Hi.
was very interesting. Thank you, Dr. Anish Kumar, for sharing your knowledge and experiences in
Thank you.
DNA sequencing technology. And this is the time for this discussion. You can type your participants can type your questions in the chat box or unmute your mic and directly you can uh, talk to the sir.
Yeah, I see a question from Licinta. So the question is, what are the major contributions of DNA sequencing in COVID-19 vaccine preparation? So, well, I don't really work on, COVID-19, uh, but, mine is almost like a layman's, comment here. It's not really an expert opinion. So the next-gen sequencing was used actually to identify the mutations in the COVID-19 genome in different isolates. You have so many uh, uh, you know uh, isolates now all over the world, and that has been uh, isolated and sequenced at full level to identify these mutations. And these mutations are important for several purposes. One is to understand the virulence of the virus. Like certain mutations can increase the virulence, certain things can decrease the virulence. And you already have the data how that particular isolate is associated with very virulent version or not. Then you can try to make connections like that. So another aspect is mutations affect the vaccines, right? Vaccines are the antibodies or T cells that recognize specific epitopes. Okay, that and mutations actually can change the epitopes, which can again lead to the success or failure of the the vaccine. So That's a general comment I can give, because I don't really work on this area, so I don't have a very good depth in that. Yeah. Okay, okay. So somebody noble. If I want to find alterations present in a fresh sample, which NGS platform is better? Can you please explain possibility of whole gene exome sequencing? So okay, yeah, that's a good. So the idea is, if I understood the question correctly, if I want to find alterations present in a fresh sample, that means if you have an individual, and you want to see what are the kind of differences in the DNA sequence of that particular person, you can use any of the NGS platforms available. So, but it also depends on which one is used most. Illumina is used most, but any of the NGS platform will be good for that. Basically, you need to do a whole genome sequencing. That's one way to go for it. And can you please explain the possibility of whole exome sequencing? So exome sequencing is something where you are looking only at the exons. That means you are looking only at the protein coding regions. Okay, the majority of the DNA in our cells, now genome is not protein coding. Okay, so people think that protein coding, they are the most important thing. And also to sequence the whole genome is much more expensive than doing just the exome. That means only the exons, because exons are only a fraction of the total genome. So that's why people used to do exome sequencing, just to identify mutations or polymorphisms within the protein coding genes. So that is good. That has led to identification of large number of polymorphisms associated with diseases. But that way you are losing out information on the other regulatory elements which are not part of any exons or protein coding genes. So that's the problem there. But again, it's always a balance between the expense and the information. uh, Doing whole genome, again, finding differences in whole genome is also computationally also expensive. It's more complicated. On the other hand, exome is much easier. So yeah, that's how it works. It actually depends on the particular need. You have to decide on what is the methodology that you need to use. Any other questions?
Hello? Any other queries? Sir, there is a question from Disinda. What are the major contributions of DNA sequencing in COVID-19?
I think I answered that.
Okay.
I think I answered that. I did answer that.
Excuse me, sir.
Yeah.
Am I audible?
Yeah, you're audible.
So actually, is there any difference in the efficiencies of the sequencing techniques?
Okay, that's a very, very, very good question. I've shown you some of these slides, right? How we actually read the sequence. You're looking for the fluorescence, right? And in a very, very, very crowded field. Okay, so there are issues with that, and that varies with the platform that you're using. Like in case of Illumina, it may not be really, there can be overlaps if you're not really careful about the concentration of the DNA that you're using. That means if you're using much higher concentration of DNA or the library than you need to use, you can actually have overlaps between the clusters, right? So that kind of problems can come, and always there is an inherent error associated with the base calling. So each system or each platform has its own advantages. Okay, like uh, in case of when we come to pyrosequencing, okay, in that case you are adding one nucleotide at a time, okay, and there is no blocking there, blocking of 3' end. So in cases where you have multiple, you have a repeat or a, oligo A, or 3 or 4 A or T or C coming at the same in contiguously, in that case, it's going to be difficult because there is no blocking there. So if you're adding A, and if there are 4 A's continuously there, it's going to add all 4 A there. Then you have to actually make out from the intensity of the signal, how many A's are there. So that you have to do computationally. Okay. And also if you're in case of Illumina, if your clusters are not formed well, there can be problems or if they're overlapping, there can be problems. So there are very complicated computer algorithms to actually make that error as less as possible, but still error is part of it. Okay, there is no other way. But they also predict what is the reliability of this. That means when we get the sequence, okay, from you get this millions of sequence, right? So everything is associated with, for each base is associated with a quality score. Quality score means whether it is like, is it 99.9999% reliable, that kind of quality scores are associated with it. But of course there are errors, and like it depends on the system. The like in case of pyrosequencing, you'll have more of indels, insertion and deletions. In case of Illumina, there are other kind of errors that can come. So how we address it is mostly computationally, and by predicting what could be the real one.
Hello.
Any other question?
Hello. Any other questions or queries? Okay. So okay, we can conclude the session. The session was very useful and fruitful for all the participants. Thank you, Dr. Anish Kumar. Then, on behalf of Department of Biochemistry and Marathanesius College, I express my sincere gratitude to you for spending your valuable time for this webinar. And I am sure each and everyone that participated in this webinar have got a very much of knowledge about the DNA sequencing technology. Then I would like to express my heartfelt thanks to Dr. Shandy Evera, our principal, for her valuable support. Next, I thank Rusa for the financial support, and special thanks to Dr. Julie Jacob, Head Department of Biochemistry, Elizabeth Jacob, Paul George, and all other uh, colleagues of biochemistry and microbiology, biotechnology departments of Marathanesius College. Then, last but not the least, I would like to thank all the technical team and the participants for the support throughout the webinar. Thank you all for your kind cooperation. Thank you.
Thank you.